Good afternoon, students. Welcome to today's biology class. Just to recap you that what we have learned in the previous class, we have started with the introduction of the chapter, that is life processes. What is the life processes? We have discussed that first. The meaning of the life process is like the different types of activities that the living organisms does. Then we have discussed about what is the difference between living and non-living thing, on what criteria we differentiate between them. Then on these criteria, then we came to the topic on the different life processes. There are four. One is the nutrition, then it is the nutrition and the digestion, then respiration, then transportation, and then excretion we had discussed. And then we came to the nutrition. We divided on the basis of acquiring the food for them. We divided into two categories, that was autotrophs and heterotrophs. We kept the plants and some bacteria in the autotrophs. They prepare their food on their own by using the inorganic components. And then then heterotrophs are those which are dependent on the plants and other sources for their food directly or indirectly. Okay, so then we came to the autotrophs in detail. We studied that how this photosynthesis takes place. What is the anatomy of the plant that makes it enable to make the food on their own? Okay, so we jot down that there are basically the inorganic components are water, sunlight, then carbon dioxide and chlorophyll. These are the four important constituents, or you can say the components, for the autotrophs to make the food. Okay, and then so we have shown you, I have shown you that how the um, carbon dioxide enters in the leaves, and how the sunlight is trapped by the chlorophyll present in the chloroplast, and then how the sunlight solar energy transfers into the chemical energy by splitting up the water molecules, and the carbon dioxide gets attached with these hydrogen molecules to form the glucose and the form of the food in the plant. So I have shown you the internal structure of the leaf also. There are few different types of the cells, and each type of the cell has its own feature in the process of photosynthesis. Okay, this much we have discussed. And now, just to tell you to check whether you have written the correct answers of the practice session 1, I gave you 10 very simple questions based on the lesson that we have done in the previous the portion of the lesson that we have done in the previous class. What was the first question? The first was the organisms having parasitic mode of nutrition. What are they? Parasitic mode? Yes, parasitic mode is, you can add the plasmodium. Okay, then we can add the fungus. These are the parasitic one. You can write any one. Okay, and then second one was, name two inorganic substances which are used by the autotrophs. Just now I have discussed, one is carbon dioxide, other one is water. What is the mode of nutrition does a fungus has? Fungus is a saprophyte because it feeds on the dead and decaying components of plants and animals like molds, mushrooms. Next, question number 4. What is the gas used in the photosynthesis? That is carbon dioxide. Fifth question. What is the gas name produced during the photosynthesis? Which gas is produced? Very, very, very important for us. What is that? This oxygen. Because by the process of photosynthesis, carbon dioxide is used up and the oxygen is given out, which is used by everybody, which is a very, very important gas for all of us. Next, question number 6. Where is chlorophyll mainly present in plants? Where is chlorophyll present mainly in the plants? Chlorophyll is present in the chloroplast, basically in the leaves and the tender stem. Okay, we have discussed that where is the maximum in the mesophyll cells. It is present in the leaves as well as in the stem portion. Next, question number 7. Which pigment that absorbs the solar energy? What is the pigment present in the plant that absorbs or traps the solar energy? Chlorophyll. Question number 8. Name the type of the cell present inside the leaf. The types of the cell present in the leaf is epidermal cell, then we have mesophyll cell, palisade and spongy, then we have guard cells. These are the mainly type of the cells which is present inside the leaf. Question number 9. Define life processes that you have already written in your notebook. I have given you the proper definition of that. So you have written it. So the type of metabolic activities done by any living organism to survive on the earth is called as a life process. Last question. On what criteria a living thing can be distinguished by a non-living thing? What is living thing can breathe, non-living thing cannot breathe. Living thing can show internal as well as external movement, but non-living thing cannot show any kind of movement. Next, living things can reproduce. Non-living thing cannot reproduce. Okay, living things have life. Non-living things does not have any life. So this was just a discussion about the questions that we have done. And children, all these practice questions you are going to do day wise, and you will keep all these papers intact with you so that after the reopening of the school, you all have to submit date wise. And for this is for the evaluation purpose. Okay. Now today's lesson we are going to start with the stomata. The topic is stomata because we have end up with the anatomy of the cell of the leaf where the cells were shown: upper epidermis, lower epidermis, mesophyll cells, and in the lower epidermis the gaps was there with the guard cell. Now between the guard cell is enclosing a space which is called as a stomata. So little more detail of that we are going to do it today. Stomata. Let's go with the definition of the stomata, children. Okay. So in the simple language, we say that stomata are the tiny pores present in the leaves through which exchange of gases takes place. So what are they? They are the small pores, or you can say that holes type of thing in the lower, maximum it is in the lower epidermis, but some of the stomata is also seen in the upper epidermis. Now children, we will be conducting a practical of making a slide of the leaf's cross section to show the stomatal opening of the leaf. How many are there in the upper epidermis and how many of there are there on the lower epidermis. And each stoma has a pair of guard cells. What is stoma? Stoma are those that gap which is present in the between the guard cells. When water enters the guard cells, it swells and the pores opens, and when the guard cells lose water, it shrinks and the pore closes. Now, on the screen, you have many diagrams in front of you. The first diagram, this I am talking about. When you will make a cross section of the leaf, you will just take out the peel of the epidermis of the leaf and you will just spread it on the slide with the help of a stain color, and you will put it under the microscope. This type of structure you are going to view. What is this type of structure? These are the guard cells, light color, and the central portion which is dark green color is showing the stoma. And these uneven structures that you can see on this page is the epidermal cells, which is enclosing, which is holding the guard cells intact in the place. Okay. Now once we will just show you this particular area in this surface view. This is the upper surface, like from the top, when you will see how it looks. You can see that this brown color portion which is enclosing the green color portion is called as epidermal cell. Okay. each epidermal cell has a nucleus. You can see that each epidermal cell has a nucleus. Now inside the epidermal cell, the green color bean structure is called as a guard cell. They are guard cell, and inside the guard cell you can see that there are green colored dots are also present. So these green colored dots are called as the chloroplast. Now what happens, and you can see that there is one very light thick portion in the guard cell, inner part, that is called as the inner wall of the guard cell. That is very little thicker. It is thicker than the outer wall. The inner wall is thicker as compared to the outer wall. So what happens and how this opens and closes, that will come later. Let's see the cross section. Suppose we cut out from this portion. If we cut like this and then we see, you can see that the epidermal cell has a nucleus, the guard cell has nucleus, and the guard cell has green chloroplast also, and the inner wall you can see that how thick it is. Okay, now what happens, how it opens, now what happens, this is the lower epidermis. What happens when the water is transferred from the epidermis, H2O is water, when the water is first absorbed by the epidermal cell, this epidermal cell absorbs water, and then here the water concentration is more as compared to the guard cell. So what happens, the diffusion process will take from epidermal cells to the guard cell. It will come towards the guard cells. So the water is getting accumulated in the guard cells. So what happens, the guard cells swells up. It swells up. So what happens, it stretches. It stretches, so inner walls also stretches and the stomata opens. During the hot day, what happens? Whatever amount of water is being retained by the guard cells is sent out to the epidermis by the process of transpiration. So in that case, in this picture, you can see the water is going out to the epidermal cell. So what will happen? The amount of water, it is more in the guard cell now, which will be going to the outer, because here the concentration is higher and here the concentration is lower. So it will, arrow you can see it is in the reverse direction. The arrow is in the reverse direction. So what happens? The water moves towards outside. So the guard cell shrinks. When the guard cell shrinks, automatically the gap between the two guard cells also closes. So the stomata encloses. So this is the mechanism. If the question is asked, how the stomata opens and closes, or how the guard cells ensures, or how, what is the mechanism the guard cell does to open and close the stomata. So you can say that when the water is absorbed by the epidermal cell, it transfers the water from it to the guard cell by the process of diffusion, and when the water is absorbed by the guard cell, it swells up and it pulls away the gap between the guard cell and they opens. And how they closes? During the transpiration process, the water is transferred from the guard cell to the epidermal cell. So this way there is a decrease in the amount of water in the guard cell. So this makes the guard cell shrink. So what happens? The gap between the two guard cells also reduces and it shrinks and it closes the gap. Okay. So this was the mechanism. Next, this is the very close view of the slide. You can see that. Okay, how it will look. Now when you are going to make this slide in the lab, your slide should be like this. The different stomata should be visible. And if it is not visible, that means you have made some error. So we will practice this. This is a very important practical for your class 10 board practical exam. Now you can see that this is a very closer view for it. You can see that this is the opening mechanism. Water is coming from the epidermis to the guard cell. So what happens? The gap is increased. And when the water goes from the guard cell to the epidermal cell, then it is closes because the cell shrinks and the water is not present inside the guard cell as much as it was present when it was open. Okay. Children, everything which a science which is said in the science has to be proved. We said that without water, plant cannot do photosynthesis. Without carbon dioxide, we cannot do photosynthesis. Without chlorophyll, photosynthesis impossible. To prove these facts, we have to conduct an activity. So scientists have given an activity and they have proved that each of the ingredient is an important part for the photosynthesis. So first we will start to the activity which is shown, which is there in your book, that how we prove that chlorophyll is important for the photosynthesis. Okay, so next we have the activity which I am reading first, then I will tell you one by one step. Now, what is the aim? What we are going to prove? To show that chlorophyll is necessary for photosynthesis. This entire activity is given in your textbook. Children, have a look. Okay, and then that is why I've given it to you in the points wise. In that book, it is not given in a very clearly point wise. It is not given. It is given in the kind of a paragraph. And this activity based question also comes in the board exam. This year also in the board, this particular experiment was given and the question was asked. Okay, so first step is take a potted plant having a variegated leaves. Variegated leaf means, children, a kind of a leaf which is not completely green, which has some kind of spots, which has white spots, yellow spots, red spots, which is some portion are green and some portion are of different color spots. Okay, so we will take like croton plants, decorative plants, which is there in your garden and you must have seen that. So we will take such kind of pot which have this type of leaf. Then we will keep that pot inside a dark room for 3 days. Now, dark room means inside the house where the sunlight is unable to reach. We will keep that potted plant in the dark room for 3 days. Why? Because without sunlight, the plants cannot do food preparation. So whatever the food was stored in the form of starch is being used up. Next step, after 3 days, bring that potted plant out in the sunlight for 6 hours. So what happens when we suddenly will bring to the sunlight, so that it can do some part of photosynthesis in the sunlight. Third, take a leaf. After keeping in the sunlight, you just pluck one leaf from the plant and then on that leaf we will keep a paper. We will keep a paper, and what we will do on the paper, whichever portion was green, just mark on that outline of the leaf. So that green color portion we will mark it. Okay. Next step is that after marking, take out the leaf and then you put it in a boiling water. Take a beaker, boiling water in the beaker, and we will put the leaf in the boiling water. This will make the leaf soft. Now, then we will take out that soft leaf. We will take a small beaker, or you will take a broad test tube. We will take some dilute alcohol, ethyl alcohol, ethanol in that test tube, and then we will put that leaf in the alcohol, and then that test tube we will keep it in the boiling water test beaker, in like it's a water bath. We will take a beaker, water is there, and then inside that boiling water beaker we'll keep the test tube, so that, and in the test tube what is there, the ethyl alcohol is there. We'll keep for 5 minutes. We will keep inside the water bath. What is the role of the alcohol here, children? The alcohol has an ability to decolorize. It will take away all the chlorophyll from that particular leaf. Chlorophyll will be washed away by the alcohol. So if we will remove the alcohol, we will remove the chlorophyll with the help of an alcohol. Now, once the decolorization process is done, we will take out that leaf, we will wash the leaf in the water, and now dip it in another solution, that is the iodine solution. Everybody is aware about the iodine solution. Where do we use this iodine solution, children? Yes, to test the presence of starch in any food item. If you take a potato, we'll put some iodine solution. Iodine is generally brown color solution. But when it comes in the contact of starch, it converts into blue and black color. So now we will dip the leaf in the iodine solution. What will you see? Some portions of the decolorized leaf are converted into blue-black portion. And you will be surprised to see that now you take out that paper that you have marked the green color portion. You will see that exactly those areas have converted into blue-black. What does it show? It shows that in that 6 hours, when that chlorophyll came into contact with the sunlight, it trapped and it has done the process of photosynthesis. That means broken down the water molecule, and it reduced the carbon dioxide by combining the hydrogen molecule to carbon dioxide, and it changed into glucose molecule. So those areas and the glucose molecules combined together, they converted into starch. So the green color portions were having the starch, and that only showed the presence of starch by adding the iodine solution. So this proved that if that portion, why the other part of the leaf was not showing the blue-black color, because the portion which was having the spots, white spots or yellow spots or red spot, they did not show the starch presence. Why? Because in that portion the chlorophyll was not present. So this proves that chlorophyll is an important component for the photosynthesis process. Children, so today I end up my session here. There is a practice session 2 for you today. Questions are there. Please do it. Then in the next session, I will ask whether you have completed it or not. Thank you so much, children. Have a wonderful day.